Yeah, I think we can start, no? Yeah? So, well, thank you so much for coming. Today it's a pleasure to have with us Gabor. He's a professor from the Texas State University. Well, we met like, 4 or 5 years ago in Oviedo, and then we have been collaborating for several years. Then I went to Texas last year, and they took out of care of us, so I think that she's going to enjoy her stay in Seville. Caitlin is a physiologist working on behavior and trying to understand also the consequences of anthropogenic disturbance, mainly on leaf-bearing and amphibians, and how is the link between the different mechanisms underlying the responses and different life history traits. So, well, I think with this you can just start. Thank you so much for coming, Caitlin.
Thank you, Pablo. Well, thanks for coming. I hear it was a holiday yesterday, so, I mean, I know it was a holiday, but some people might still feel that they're on holiday. And before I, I'll just use this. I think, is it frozen? That's not what I want. I think it's in a weird setting. Oh, that now we went to that. Okay, so before I move forward, I wanted to thank Pablo, and we have our own arches because, you know, in Texas you can make anything you want and just make it big. So Pablo came to visit us, and it was been really great working with him, and so I was very excited to have the opportunity to come here and see where he's doing his work and talk about some of my work and hear what everyone else is doing. And in many cases what I hear is that we're all doing something similar because we're all seeing major declines in species or whatever reason, but effects of climate change on organisms at all levels. And I don't know why this is. So this is a picture of Austin, and I didn't ask, maybe it's not on. There, on. There, now it's on. And there. So this is a picture of Austin, and it's also a city with a river running through it, and that affects our research. There's actually, unlike what most people think of Texas, there's a lot of water in Texas, and more so than in these regions. But we know from that urbanization is associated with population declines, non-human extinctions, and reduced levels of biodiversity. There, now it is working. And this land use conversion, especially where we live, has major impacts on streams, because if there is no development, then most of the water from rain, only a little bit will run off, and most of that will go into the ground when there's 0% impervious cover. On the other extreme, in the cities like Austin, there's so much cover from roads and buildings and just general development that a lot of water ends up running off in higher impervious areas with higher imperviousness. Okay, and so the outcome of water running off is that what's known as urban stream syndrome. Basically, so much water is running off of the impervious cover into the water, it changes a lot of components of the streams. And what we see is that freshwater fauna are declining 5 times faster than terrestrial fauna. So a lot of the work of the research I cite is based on birds, and they're always thinking about either in the air or terrestrially, and a little less so in terms of aquatic organisms. But these aquatic organisms are experiencing increased volumes of stormwater runoff, more nutrients from that runoff, and exposure to fertilizers, pesticides, and other contaminants. And for example, in Texas, where despite a lot of water, There's also extreme droughts, and yet people want to have a garden full of grass and use pesticides and fertilizers to keep it going. And what I noticed to connect what we have to what you guys might have is that when we were in Granada, it was so nice seeing the water, let's see if it won't do it on, being channeled away from the roads and into, they're actually using it and putting it back into the mountains. So we went on a free tour, and the person explained all about how water was saved and reused and used. And this does not happen in Texas. Our water's just everywhere, and it's a mess. And we have a lot more water. There's, I guess in centimeters, there's probably about 3 times more water rainfall in the area of Texas that we live, but we don't do anything, not nothing, but not so much where we channel water and leave it out. So the impacts of urban stream syndrome are really extreme. And the outcome of urban stream syndrome is increase of tolerant species, and I'm sure there are mosquitofish here too introduced worldwide. And there's a decrease of sensitive species. These are our local species. So again, central Texas actually has a lot of water relatively despite droughts. This is a Texas blind salamander. These are found underground in caves. So we have a limestone-based system that's similar to, apparently there is some also up in Oviedo where I am now, and the Texas, this fountain darters that are only endemic right in San Marcos where my university is. And so we have a decline in these species and tons of these that no one really wants. And so I have multiple big picture questions that I want to talk about today about the effects of urbanization. First, what are the consequences of land use conversion on behavior and glucocorticoid stress response in multiple systems? How does land capacity, how does the capacity to cope differ across these different levels of land use? So here are all these streams running through the center of Austin. I don't have the map to show it all. I will in another one. And this is more red is the more urbanized areas, and then the green are the less urbanized areas because they're not developed. And then there are some big lakes up here, and these are some examples of populations where I'm collecting. So we have all this urbanization. We want to also know what are the fitness consequences of the modulation of stress hormone in response to this land use conversion, how coping capacity differs, and whether populations can respond to selection associated with environmental change. And since, again, we have a broad group of scientists I want to at least introduce, I know that, of course, Ivan and Pablo are physiologists, so their groups are aware of it. But the glucocorticoid stress response, generally, what we see is that glucocorticoids, the main one in amphibians and fish, primarily in amphibians, is corticosterone, and fish is cortisol. And there we see this stress response released within about 3 minutes after a stressor is perceived by the brain. And so what we expect at baseline are for whichever glucocorticoid is of interest, that when there's a perceived stressor, the organism will show a stress response, and this can aid in energy release and the immune system response, but we also expect the organism to recover to their baseline levels after that stress response. But a couple other things can also happen in more stressed environments, such as if there is chronic stress, they may not show a stress response to a perceived stressor, and so it stay what we might consider down at the level of baseline, or the glucocorticoids could stay up high in response to stressors. Generally, at this point, they're probably about to die. But we need to recognize that it may not be changing, in other words. And so what I thought about then is how this stress response and recovery, or people are thinking about, is how this might aid in their ability to respond to change, their capacity to cope with changing environments. And so what you might see is that if we look at their baseline stress response and recovery of a given organism, and whatever their glucocorticoid is, and we think about whether they're living in potentially what we might consider like a natural forest, an unstressful environment, or a stressful environment like outright in a very urban area. And that if they were in an unstressful environment like here in blue, and they had a regular glucocorticoid stress response, we would get a high stress response and a quick recovery from that stress response, so they're not constantly having the cost of being in a stressed state. And so we're calling this the stress response, of course, and this recovery, that negative feedback, getting back to baseline. But maybe in a moderately stressful environment, they will still show a stress response, but may not recover as quickly. So the slope of the lines are telling you something. And also that the baseline cohort may start at a higher point due to the level of stress in the environment, maybe. These are just predictions. And then if they're in a very stressful environment, or an environment more, what's a better word is more unpredictable, then they may have very high baseline cohort and not really show a stress response and recovery. This could be indication of their capacity to cope, and having this ability to respond with a quick stress response and a quick recovery would be aid in their survival and reproduction. And then there may be costs to this sort of response. Okay? So we expect the ability to rapidly turn on, that's your stress response, and also recover from those as the least costly. So if we then look at that in terms of reproduction, you might have higher higher fitness. And what we know is it's not well understood, or many people are trying to understand, why some species are able to adapt to urban living and others perish, like those mosquito fish. And so the outline of my talk is that first I'll talk about the role of land use conversion on coping capacity in common toads. This is a handful of toads, tadpoles, and the consequences then of urbanization on coping capacity, behavior, cognition, and fitness in mosquito fish. So there's two parts to my talk. This first part is work that I did for a Fulbright in Hungary, and in collaboration with my Hungarian collaborators at the Lendület Institute, of evolutionary ecology, which still exists, except for that they took the science out of the academies of sciences in Hungary when I was there. And so this has changed a little bit, but Veráč Bokany and Nikki Uhi, which I cannot say her name even though I'm Hungarian, were who I was collaborating with, and then my partner Andrea Asbury, and then Jaime Bosch from IMIB, who I've been known since '97, and he ended up doing work on the ranavirus, or BD, except for we had none, and Barbara Thomsova. And so this was part of the Hungarian Academy of Sciences. And Bufo is great to work with because so much is known. The group I worked with in Hungary, they have localized declines observed. This species is philopatric, so they're going back to, they're always exposed to the same landscape and habitats because they're going back to the same ponds. It's known from the research group that I was with that PAHs, the polycyclic aromatic hydrocarbons, and bisphenol A increase with urban land use, and organic chlorine and triazine pesticides, because these are in more agricultural areas, and sex hormones increase in those with agricultural land use. And that these toads have lower body mass and development rates in the agricultural and urban ponds. So we had a really good starting point for this research. And we asked whether land use conversion affects bufo tadpoles' ability to cope, and what was the contribution of water pollution to that stress response. So this work was all done, well, was primarily done in the field. Here's Nikki in the field where we're collecting hormones at this pond. And of course, by chance, we met a Hungarian who spoke fluent English and was an engineer, and is so interesting, fishing, and what you get to experience when you're in the field. And we want to know whether the consequences of land use conversion on stress are plastic or genetic. So we have two components to this research where we consistently use CORT as this biomarker for stress from these anthropogenic stressors. And so we categorize ponds based on these 500-meter buffer zones. So we have 3 ponds that we went to. In blue are the natural, so you can tell that's a pond in the natural area. These are actual pictures from the sites we use. And then 3 that were from agricultural areas and 3 that were from urban areas. The one you saw, the first one, was one of the natural ponds. And because we want to look at whether it was genetic or plastic, we also collected eggs from 9 ponds. So I understand Rafa's how hard it was. That was just 9 ponds. We had to find 2 new ones to even work with because 2 dried before they've ever dried before. So we get the eggs and we brought them back to the lab. And when they hatched out, we placed the tadpoles into 54 mesocosms with 6 replicates per pond for 3 weeks. So these are the mesocosm, and these are my children when we were cleaning out. The tadpoles got put back into the ponds that they originated from after we worked with them. And so from some of those tadpoles, we obtained hormones from 3 tadpoles per mesocosm. And this is how we were able to tell whether in the environment versus their CORT response in the environment versus the mesocosm, whether it was plastic or not. And then in the field, we collected tadpoles, 18 per population from these 9 ponds, and placed them in beakers of 100 mils of spring water. So we did this for the ponds and the mesocosms. And we placed them in here for 1 hour, and we had the following treatments. At baseline, we catch them from the pond or the mesocosm, and they go pretty much directly into the beaker of water. And they sit there for 1 hour, and that's baseline. And then we put it in quotes because, yes, you still catch them, but they're all sort of similarly handled. And then we agitate them for an hour. So this is us all shaking every minute for 1 minute every 3 minutes, shaking the tadpoles. So that's like water coming in from the land just when it rains and just disturbing the whole pond. So they're getting shook in that way, and that's how we measure their stress response. And then they get to recover in a 3rd beaker for 1 hour. So 3 different samples of water. Then we remove the tadpole again, and as we say, we actually take the bath water instead of the baby, and we then can measure waterborne CORT assays. So we can do this for adults or tadpoles. This is at Jaime's house in Spain, these tadpoles. And CORT is primarily assayed in especially larger animals via plasma or whole body for tadpoles. And this method is non-invasive, so I've been talking to Katerina about it, and we're using enzyme immunoassays. And it's based on the hormones released from the gills, urine, and feces. And then we know there's a correlation between waterborne plasma CORT and their circulating levels that was validated in frogs and salamanders. And it's viable, as you see, in the field. So we don't have the limitation of having to only do it in the lab. And it's considered less stressful. There's less handling of them. And finally, what I think is the most important and interesting is it allows for repeated sampling. So before this time, we couldn't get the coping capacity of baseline stress response and recovery because each tadpole had to die for a sample. And in terms of what we found, here we see we're looking at CORT across the habitats. And this is the corticosterone release rate since it's over a period of an hour. And we have the baseline stress and recovery. So remember from my image of we're expecting some kind of level, lower at baseline, higher at agitation and recovering. And then we have the data from the ponds and the data from the mesocosms. And these are the averages across 3 populations for each color, with, again, blue being urban, green being natural, and brown being agriculture. And then we see the variance around each one from the 3 populations and all the individuals within it, and then asterisks showing that the baseline for the urban population court levels was significantly higher than for natural and agriculture, which were not different. And then we can look at the slope of the curve, and we see that the slope, there's a stronger stress response in agriculture than in natural ponds or urban ponds, and then the highest court levels in these urban ponds and the quickest recovery. Whereas, remember, we're asking whether their response is genetic or plastic. So when they all got brought back to the lab, tadpoles from, eggs from the field and the mesocosms, we don't see any difference across those, and nothing that looks, and in fact, this is also all their court levels are this much lower than in the ponds in the natural environment. So what we can conclude is that they have a higher stress response, that slope, in agricultural environments. They had fastest negative feedback actually here in the agricultural environments. They had overall higher baseline and stress court in the urban environments, and the fastest negative feedback in the urban environment. So remember, I predicted that there might be slower recovery in urban environments, but we're trying to see how they're coping, and Bufo is doing relatively well compared to other species. Maybe they're coping with these environments. So we see evidence then based on this response for phenotypic plasticity and not for microevolution. Sorry, it's a Mac to PC thing, but you can get what I'm saying. And then we also talked about, we knew a lot about the water quality itself. Is it due to the water per se? And we looked at the total number of corticocorticoid disruptors. So it might change because they can absorb cort. That's work being done here too, through their skin, and their stress response based on the 3 different pond types they were in. And overall, cort increased as the number of corticoid disruptors increased in the environment, but there was no effect of the different pond types. So water pollution levels did elevate stress response, but it didn't change by the type of pond. So what we can conclude is that there is an effect of urbanization on water quality. And this is from not primarily from this work, but from other studies too, and that the coupling of the stronger stress response and stronger negative feedback in these habitats is highlighting the importance of rapidly turning on and off the stress response as a mechanism for coping. So these urban populations were having a high stress response and that quick recovery for coping with that anthropogenic change. So the urban and agricultural populations may suffer most from the land use conversion. As we see, they're showing the strongest changes in their coping capacity, or stronger responses in terms of coping capacity. So now we shift to live-bearing fish and asking similar questions, and each system has sort of pros and cons of working with them. And here, we're working with students in the field in Texas. So this is, again, my partner, Andrea Asbury, undergrad Pilo, Robert, Alex, all undergrads, and a previous master's student collecting these fish, looking at the consequences of urbanization on coping capacity, cognition, and fitness of mosquitofish. So the fitness component is what we're missing in the frog work, and this is nice to do in this system. So the western mosquitofish are naturally occupying a wide range of aquatic habitats despite being introduced worldwide. And they're live bearing, so they here are within the females, they give birth to live young, and we see those eggs developing over time. And I'm sorry that I can't figure out where to sit for you. And they're invasive and spread over 40 countries, but they actually are native in Texas. And the questions we asked was, how do female mosquitofish persist despite land use conversion? Remember, I said that there's a decline in native species, but a persistence of these more tolerant species. If they're in 40 different countries, they're obviously we know they're a tolerant species. And just to give you a sense where we live, in Austin, you know, the rule here is was the same in Austin. The tower at the cathedral is the tallest in Sevilla. In Austin, the capital of Austin was the tallest building until about 2005, when they decided to stop following that rule. And this was the first building to get taller than the capital. Then 2017, this is what it looks like, okay? So we wanted to see how, and there's a river running through Austin and streams all throughout. The fitness consequences of land use conversion, how cortisol release rates change, look at their coping capacity, their relationship between coping capacity and fitness, that's what we were missing before, and how this affects cognition and behavior, which we also lacked. And to give you a sense of what it's like, we don't have these nice little channels where our water runs off. This is in my mom's backyard that doesn't usually have water there during a very big storm. So we don't have any capacity for where our water goes. It just washes right through. So this is why urban stream syndrome is so important. What we see is, again, as impervious cover increases, the amount of runoff increases. This alters the channel hydrology, so there's just these straight sides. It's hard to get up out of the water, and there's hard to find places to hide. So it changes and decreases the amount of physical habitat, and then affects negatively biodiversity. So we can look at the percent, and so what we're interested in is how mosquitofish have altered their stress hormone and life history and cognition and behavior with urban stream syndrome. And the study sites have different percent development. So here's Central Texas, the capital of Texas. And these sites are there, right there and there. And these are, in parentheses, is the percent development. So in this green area, 1.3, barely developed. Upstream watershed. In this 1.5, but here where you see this white dot, everything upstream, you can't see the stream there, is 51% developed. Now that number is 69, and probably higher because that was actually, now is 2020, I think. So you see there's a lot of range of development in Austin, and this is agriculture, and this is hill country. So the water's flowing down. So if we return to this prediction of coping capacity, and we think about it here, we see an urbanized stream. We can think about it in terms of stress response, negative feedback, and fitness. And in a rural environment, now that we can measure fitness, we would expect that the faster their stress response and the faster their negative feedback, the higher their fitness, because the less cost of this energetic production and importance of glucocorticoids, which are preparative yet also have costs. And that maybe in an urban area, we might see fitness highest when they have a high negative feedback, so they're just decreasing their response to stressors. And what we'll come back to is what we found actually is going on with mosquitofish. So these are our two main predictions of rural versus urban response to the environmental change. So we collected females from 6 populations for 2 years. We have 4-ish years, I guess 5 now, of data that I will not present here. This is published work. And since this is very similar, we placed 20 fish each individually into their beaker of water, 100 mils of water, and they would sit there for, we only have to use 30 minutes with the fish, take them out, we put them into another beaker. Here we agitate them every other minute during a 30-minute period to get their stress response, again, of like water rushing into the stream because it's flowing fast. And finally, their recovery time for an hour. This is another stream of one of our populations. And then they're euthanized because they're live-bearing fish. We can count the number of babies. And they have multiple broods in each year, and they only live about a year. So this is one portion of their life, but they only live about a year. So here's what they look like when you open them up. Different developmental stages are seen here, and we dry the female and their eggs to measure. We count the number of embryos in a brood. This is their fecundity. And we obtain the dry mass of the fish and their brood to get an indication of, or to know their reproductive allotment. And just to put this in the same sort of way of my initial predictions, we have cortisol on the Y, and I covered up, because you said on the video I didn't notice, on the X is baseline stress. Here, baseline stress and recovery, because in the first year, we did not yet think of measuring recovery because the field has been changing a lot, and thinking about just baseline used to be what we thought about. Is it high, low, or where is it? Then how they respond to stress, and now we added in how they're responding to those, how they're recovering. So this is the percent development, and we see overall that the baseline in this low, very rural area is very low compared to in this very urban area. But we see, for example, in this urban population, a very strong stress response and recovery. So overall, you can see in these, and also urbanization is considered, an area is considered urbanized when it's over 20% development because of the effects on urban stream syndrome, I should have said earlier. So in this area with 20% or more development, we see a much higher cortisol levels in these more urbanized populations, and a stress response in all of them, but not as much of a recovery necessarily. In terms of cortisol and urbanization, we found a positive correlation between urbanization and baseline cortisol. So baseline cortisol, urbanization, and we see the increase as urbanization increases. We also see here is the stress response and percent urbanization, that they have a reduced stress responsiveness in these more urbanized ponds or streams. And finally, that negative feedback increased. Remember, we see more of them that were actually showing a strong feedback, and this was over 2 years, or that's only over 1 year, increased with urbanization. So when we then look at, for each fish, we were able to get their baseline stress and recovery, and then get their fitness, because we euthanize them and measure the number of babies. And what we see is fitness as measured by the total brood mass, fecundity, was, here's total brood mass, was greatest in individuals with this high negative feedback and a low stress response. But this was regardless of the percent urbanization. So it was not different across the percent urbanization. So it didn't fit our prior predictions of having the fastest negative feedback and the fastest stress response. So when we come back to these predictions, we see that we did not, that's what I jumped ahead and said, we did not find in these rural areas that they had highest fitness with highest negative feedback and highest stress response. And we did not see that in the more urban areas that it was the negative feedback that was most important for fitness. Instead, we see this balance between stronger negative feedback and weaker stress response, which does not correspond necessarily with what we predicted here. So mosquitofish are coping by dampening their stress response, so maybe more like this, and upregulating, though, their negative feedback. So this was stronger and this was weaker. Whereas if we look at this in relation to the tadpoles, in the common toad tadpoles, they upregulated the stress response and the negative feedback in response to urbanization. So we were looking at how urbanization affects female mosquitofish, and we see that to some extent these are tolerant species. They are coping. They're coping differently than tadpoles to some extent, although we're not seeing this clear relationship that we expected between coping capacity and fitness. And what we wondered next was, what is the effects on cognition and behavior? And here is our son in our stream behind our house with this limestone bottom, and you can see all the cutout underneath. These are the effects of urbanization, urban stream syndrome. So that is this altered hydrology, the altered channel morphology, reduced physical habitat, and maybe increased predator success. So we expect it to alter cognition and behavior. So this will be the last part of my talk, but it's really fun to think about how urban stream is affecting cognition, because it mediates this perception of the environmental stimuli, and because we see changes in cognitive levels across across different types of environments. So we might predict a response to urbanization. And the process of cognition consists of learning, memory, or cognitive skills, some component of learning, memory, and problem solving. And these then can modulate their behavioral response, in addition to being able to measure risk taking, which we'll talk about, which is this willingness to explore novel habitats. So we say urban stream syndrome is changing the channel morphology. And so we expect a change in the level of heterogeneity in the habitat and structure, and that potentially, based on studies with birds primarily, that in an environment with lower heterogeneity, also mice, actually, again, terrestrial organisms, in lower heterogeneous environments, the organisms will have lower cognitive skills. And so we know that there are areas like what you saw in that picture with very low heterogeneity in the environment, some with moderate and some with higher. And we looked at how this affects cognitive skills of mosquitofish. And this is my grad student, Kendall Irwin. And this is the chambers we used in the lab, where she would collect them in the field, and 2 days after bringing them to the lab, test them, so they had a little bit of time to recover from the stress. And what she did was she measured habitat complexity using optical analysis. So these are 3 different streams with the low complexity, moderate complexity, and high, and using a camera, and then using this image J, she could measure the range and put them in relation to each other of complexity, so you can see to some extent how low, medium, we categorize them as low, medium, and high. And this is her in the process of getting that data. So what you can get then is we have these detour tests that are measuring cognitive skills. We placed a focal fish in an enclosed chamber, so we were able to measure the behavior, which is the willingness to exit or risk taking. And then when we opened up the arena, and then they have this clear barrier, and they can see the reward of another female. These are shoaling fish. They like to be with other fish. But when they reach the barrier and attempt to be near another female, they can't reach her. So they have to go around that barrier, and at this time when they're going around, they can no longer see that fish because of this opaque barrier. And so what we get is we can measure risk-taking, the willingness to come out of a dark area where they've habituated, the motivation to get to the female, the time it takes to reach that barrier, and their solving ability, their ability to get around this and get to the female. So the best part about this is watching this population. Here's one fish, she's coming out. We had already opened it up. And she slowly has not yet, you see the other fish? Okay, so that's motivation time, and that was, we measure also from when we first opened it, exit speed. And this female has not solved in the amount of time we're talking, okay? This is from another population, a different population, Guggens. She's never been in that chamber before. Is that crazy or what? It's our favorite fish. And so we measured these cognitive skills, and what we found varied was this time to come out of that darkened chamber, exit speed or risk-taking. And the faster they come out, the lower that number is. And this is solution speed, the time it takes them to get. So the Guggens population is in low, and the solution speed was super fast for the whole, on average, for the population. And their time to come out was super fast. And similar to medium complexity environments, but much faster, significantly faster than high complexity environments. In solution speed, again, the low complexity populations, there was 3, were significantly faster at solving than at medium and high complexity. So low complexity populations took greater risks and were faster at solving the problem of reaching a goal. So this faster solving ability in these low complexity environments is, or compared to high complexity, and also they have greater risk taking, is actually opposite of our initial prediction. We predicted that in higher complex environments, their cognitive skills would be more developed based on results from terrestrial organisms, and we found the opposite. And that in the high complexity environments, they were actually less risk-taking. Our prediction is that in these, one reason this might be the outcome is that the predation pressure might be driving it. We don't have that data with this because we didn't know what was going to happen. This was all new work. But in these got on the wrong button. Low complexity environments, the fish have nowhere to hide. And they need to forage. So we think that when they're coming out, they have learned where they can go quickly and potentially avoid predators in those, whereas that's less required in these kind of environments. But this is one of many possible hypotheses for why we found this. Alternatively, for example, in the high complexity environments, those fish from them are being tested in what is clearly a low complexity environment, and so their response to it may just be different from their previous exposure. So does urbanization affect female Gambusia finis? We see that their coping capacity does respond and change in response to urbanization. Although it doesn't necessarily increase their fitness per se, there's other reasons, like in urbanized environments, the streams, since they're oligotrophic, meaning they have barely any nutrients in these limestone streams, there's much higher fitness in the urban areas because of runoff, so there's more nutrients in them. So that's why we may not see this relationship. In terms of cognition and behavior, we see an effect of changes in the complexity of the stream on cognition and behavior, although there's no correlation with urbanization per se. I left that out. And so tolerant mosquitofish cope with anthropogenically modified streams by altering potentially cognitive skills, their GC profiles, and having increased fitness. So together, it's not just one solution to living in an urban environment, but many. And I didn't show you fitness data directly. And so these results contribute to our understanding the mechanisms driving these species-specific adaptations, and thereby community structure in these freshwater systems associated with these land use converted areas. And where we want to go from here is expanding our system to consider gene expression and microbiome. So from each fish, also like in Yvonne's lab, you can really get a lot of information. We already know effects of urban stream syndrome on their court response, on their fitness. We're looking at population levels, but we can get their molecular response in terms of gene expression changes, gut microbiome changes, and we can look at that over space and time. Are they doing the same thing every year, or is it changing? Or are environments that were low complexity, are they doing the same thing once they become high complexity by being able to follow them over years? And with that, I need to thank, so in Texas, everything is private. So all those places that were not developed were private land, of course. And so we got access to those via Texas Ecology Laboratory, my Fulbright work, all the students in my lab over all of these years. With that, we'll take any questions.
Thank you very much, Caitlin. So time for questions.
Yeah.
For the cognitive tests with the fish from the different complexity habitats, were those animals from the wild or the offspring of animals from the wild?
I remember I said there was a good part about the fish and about, these are directly from the wild, collected 2 days in the lab. We want to look at plasticity like we did with the tadpoles. But since the good part about live bearing is they have them in them, the bad part about live bearing is they have them in them. When they come to the lab, they love to eat their babies. So we just haven't done that work yet. We pretty much have that solved now, but it was not what we did.
Thank you very much for your talk. About the fitness measure that you use, the number of babies, could you have done it with other kind of fitness?
We have reproductive allocation, which is the mass of the brood. So then you can look at life history trade-offs, but the trade-off today was that I didn't talk about that.
No, I meant not using the brood. Other fitness.
How long they live,
item.
maybe? I mean, they only live about a year and a half, so that's really, I mean, usually that's the golden standard is actual offspring.
Body.
Oh, yeah, yeah. All of there are a lot of other amount of fats, how much fat is in their body, and so yeah, there are other measures that I'm primarily a behavioral ecologist, and then I use some physiology to help out. So I actually ended on my favorite part and started on, I like all the rest, but it would be a good collaboration to have with Pablo and Yvonne, and yeah.
Thank you very much for this talk. I actually have 2 questions. So the first is very physiological regarding the waterborne part method. So we are measuring the part that has been released by the specimen into the water, and instead it's released in gills and also through feces and urine. So I would assume that in the urine and the feces it's more like part from the past, and for gills it's more like acute part release. Is there any way we can distinguish between these 2 different kinds of parts that have been saved that way?
I don't think that that's necessarily true because it's bound court, and that's part of why anything released from the blood is still released. So it's not necessarily, I know, I don't think that feces versus gills is different information.
Okay, thank you. And the second question is, you were talking about the corticoid disrupting compounds. Which are these in particular?
anything that is a steroid type shape, but this is all the stuff from the thankful of collaborating with the Hungarian Institute. I can go through from the paper and tell you, but
Okay.
I don't have on my head, top of my head, a list of those. We also looked at other non-corticoids and that are other pollutants in the water, but only the corticoids were what showed anything going on.
Okay, thank
So
you.
I'll show you the paper.
Any more questions?
Yeah.
Yeah, just curious about the predation. I mean, I imagine you've got different predator communities in these different environments impacted more by urbanization in terms of not just fish communities but also bird communities, and the responses that fish would have to avian and fish predators would probably be quite different in terms of how they escape. So is that something you've been thinking about, and what are your thoughts on that?
The predator communities? Or
Yeah, and how that
they
changes
different?
across this gradient and how that might affect the behavior.
It's something we need to be thinking about. It's not something we've yet worked on because one of the things I think about is when you go in and you collect, for me to say what are the, let's say, aquatic predators, other fish or insects, you need to say over and over and over again and say, okay, in this population we found this, and then what happens to the stream that I want to go back to every year. It's something that I've had a problem with for a long time, and being not an ecologist, I've decided to direct my hypothesis to things I can do and not the things I don't want to have happen to the environment. As to what I actually know at this moment is there are people who are doing that, of course, with fish. And the reason we came up with the hypothesis about the predators is that in more urban environments, there's sunfish, for example, that are pretty much common predators that are more pervasive in urban environments. And like we have data, where they count all the fish, just not at the exact sites we were at. In terms of bird predators, I think I don't have that sort of data, but I think they could be similarly at both, because there are some birds that are quite urban that we see in those streams, but is definitely another component I did not have a way of considering. Yeah.
Caitlin, thanks a lot for the talk. I was wondering, in the Bufo experiment, when you compare, you don't find differences in the mesocosm, but you find differences when you sample directly from the ponds. You attribute that to plasticity, but do you think that what other factors, because mesocosm, you're highly controlled, but then when you go out in the field, there could be a number of non-controlled for factors that could be contributing. Is there anything that comes to mind that could be different in the ponds that were not accounted for in the mesocosm experiment?
The chemicals. The pollutants are the first one, which we know differ across the populations based on their prior work. Predators. I think that, and the what are other organisms living in the water at the same time. So the community. Those are the major ones that I would think of.
Thank you, Caitlin. Yeah, it's very interesting your talk, because you get an integrative view between the green approach, adaptive responses. So it's very interesting. Well, I have a minor comment that's related that low populations living in low or less heterogeneous habitat seems to be higher level of cognitive recognition, right, and so. But you attribute this to because higher risks of predation or so. But have you measured other traits, or have you planning to measure other traits based that these populations are really adapted to face higher predator risk, for instance, higher speed velocity or swimming speed, or so, to cope with these predators, or other.
The swimming speed is
something like that.
Very right. Look, so this was just yesterday also, I forgot, was provisionally accepted in biology letters. But provisionally with this, like, there are definitely a lot of caveats of what we don't know. And so that student is staying for her PhD. And so, like, one of the first studies will be the part about considering if they're in a complex environment and they get tested in a complex environment. And then other components like how, if you look at exploratoriness and you look at, like you said, velocity, are those changing? And more ecological questions of communities in their environment. So for now, we know that we haven't considered everything, but it's a fun hypothesis that we're think is supported but is not tested.
Next work.
Is likely to be supported when we test it.
Okay,
yeah.
Thanks.
Thank you so much for the talk, Kathleen. My question is about the glucocorticoid dynamics. So I also bought the story that where we're expecting that if you can increase the corticosterone levels in response to stress, you are expecting that this is going to be probably more adaptive because you are allocating more energy, blah, blah, to different traits. And then if you recover faster, everything is fine. No? But my feeling after seeing your graphs is like normally what you find is a difference in the mean values between different populations, no? For example, regarding different urbanization levels. So do you still think that the dynamic is that important? Or, for example, if you have to use the corticosterone levels in different individuals from different populations, you will go more for the baseline?
That I could rely just on baseline instead of thinking of coping capacity?
Yeah. Also regarding that you didn't find clear differences in, clear link with fitness, no?
Yeah.
So.
Huh. I know, I'd have to go through them, but I don't know that, I think that you can't just, that this is the same thing that comes from you guys' lab. It's not just one component. If I just measure baseline stress and recovery, which if we call it one component, is enough. So I ended with, in the fish, that their behavior is important. The ability to just live in an environment that's been changed, and also that there's higher fitness probably just because there's more food. So I don't think, though, that I don't think I would want to just rely on baseline nonetheless, despite it not being the predicting fitness. Yeah. But it would be cheaper.
Okay. Well, we just arrived at time, so
yeah.
Thank you very much for this, and see you next time. Thanks.